Hi guys, welcome back to our YouTube channel, and today we're going to be looking at Unit 1.1 of IB Biology Standard Level course, using light microscopes. So the first thing you need to know about using light microscopes is the parts of the light microscope. And the most important parts that you need to know are the eyepiece, it's basically where you look at, the stage clips, the clips where you stick the slide onto the stage, the light source, the coarse adjustment knob, the fine adjustment knob, and the base. All of these things you're probably not going to be asked on the exam, but it's always good to know them so you know what the question refers to. So the important notes when using a light microscopes are always focusing at a low power first, using both hands to move the focus knob because using just one will misalign the stage and make your slide fall. Focus with a larger coarse focusing knob at first, then make it really sharp by using the fine focusing knob. So, we're looking at the practical of looking at the epidermis of an onion, and to do that, you first need to prepare the onion cell. So the things that you need to do are cut a 1 centimeter piece from one of the layers of an onion, and carefully peel off the epidermis from the inside, so the concave side of the layer. Place on a microscope slide and flatten gently with your finger so that there's no air bubbles under the epidermis. Add a few drops of iodine so that the cells are easier to spot under a microscope, and place a cover slip on top, taking care not to trap any air bubbles, again as I said before. Examining the cell using the microscope. First, you place the microscope slide on the stage so that the specimen is below the objective lens and so that it's visible. Plug in the microscope and switch on the power so that the light passes through the specimen. Focus with a low power objective lens first, and then use the coarse adjustment knob to bring on the slide objective lens as close together as possible without them touching. You have to be very careful because if they touch, you're not going to be able to see anything clearly. Then you look down at the eyepiece lens and move the slide downwards using the coarse focusing knob until the image is roughly in focus. And then use the fine focusing knob to adjust the focus on the image so that it looks clear and sharp. Move the slide and bring the most interesting and clear part of the specimen in the center of the field of the view so that you can clearly see it and so that it will be easier for you to sketch it. Turn the revolving nose piece to select a higher power objective lens, then refocus using the coarse focusing knob and the fine focusing knob. Adjust the illumination using the diaphragm. The scale bar is the line added to the micrograph or drawing to help show the actual size of the structures. So here you have a few examples of a scale bar. This is basically to know about which size the cell is. So this is the equation you need to know to find out magnification. So to find out magnification, you need to divide the image size by the actual size of the cell. So the image size is the size of the image you have, so the thing you have on the paper, which is going to be the biggest one, and then the actual size of the cell is the tiny value. Don't confuse those two because then you're basically not going to be able to do the equation. So remember clearly that image is what's on paper and actual size is what the actual cell looks like. So let's do one example question. Determine the magnification of the string of Margarita cell in figure 8 if the scale bar presents 0.2 millimeters. So first thing you need to do is make the units the same. 2 centimeters times 10 gives us 20 millimeters. Make m the subject of the equation, so magnification. This will give you magnification equals size of the image divided by actual size. This will be 20 divided by 0.2. This will give you the magnification of 100 times. This is quite simple, so I don't think you will have a lot of problems with that. To conclude, what you need to know is the parts of the light microscope, how to do the practical of looking at an epidermis of an onion and drawing it on paper. And you also need to know the magnification equation, which you can learn using this triangle. Thanks for watching. Please subscribe to our channel for more biology and chemistry IB content. Thank you.